GMOs, also known as genetically modified organisms, are a controversial topic in society nowadays, as more awareness is given to food safety. The practice of genetically modifying crops has been around for a few decades, but there are still many unknowns when it comes to risks associated with the consumption of GMOs. Studies thus far have either been funded by companies producing the GMOs, or have been discredited by those same companies. There are currently no labeling laws in the US for genetically modified products, so consumers don't always know the potential risks of the foods they buy in their grocery stores. That's why I chose to test samples of corn from my local grocery store for the presence of a common genetic modification known as Bt corn. As you see here, we have two cobs of corn. The top cob appears to be completely healthy, while the bottom cob looks like something out of a horror film. This is because the bottom cob is infested with an insect known as a corn borer, which works its way through the cob, killing the plant as it goes. So how can farmers avoid their crops turning out like the bottom cob? That's where genetic modification comes into play. Here we have Bacillus thuringiensis, a bacteria that produces a protein known as delta endotoxin. This protein is lethal when ingested by corn borers, because it binds to their gut walls and breaks them down, releasing gut bacteria into the bloodstream. Scientists decided to use that toxin for agricultural use. By extracting the plasmid containing the toxin gene from Bacillus thuringiensis and inserting it into the corn genome using a deactivated form of HIV, scientists created Bt corn, the name derived from the bacterium that provided the gene. The main reason that this genetic modification was created and approved by the FDA was that it kills all types of corn borers, which are extremely harmful to corn crops. The modification also rarely seems to have an effect on any other types of insects, especially insects like bees that positively affect crops. As far as we know, there's no harm to humans or other animals caused by the modification, but many studies have been shut down and no long-term research has been completed at this point. Despite this, the FDA considers Bt corn to be safe for human consumption. Additionally, by decreasing the amount of crops dying off due to corn borers, Bt corn increases harvest yields. It also gets rid of the need to spray insecticides for corn borers because the corn itself can kill the pests. So now that we know what the modification is, how do we detect it? I tested for genetic modification using a polymerase chain reaction, which detects a chosen section of DNA using a specific primer set. That DNA section is then copied over and over by DNA polymerase, amplifying the wanted section. After the PCR is complete, that section can be detected using gel electrophoresis. For my experiment, I ran two separate PCRs. The first one contained only the Cry1AB primer set, which detects the Bt gene. If that gene is present, it gets amplified during the PCR. My second PCR used the multiplex method, which involves combining multiple sets of primers into one PCR. In addition to the Cry1AB primers, I used a 35S CB primer set, which detects another type of genetic modification that protects corn crops from cauliflower mosaic virus. I also added a 16S RNA primer set, which detects a DNA segment that is found in all plants. This ensured accuracy because if there was any DNA in the sample, there should have been amplification from the 16S RNA primers. For my gel electrophoresis, I ordered my wells in the same manner on each. The first well is my thermoscientific DNA ladder. Next is my positive control, which is bird's eye frozen corn, then fresh corn on the cob in well 3, natural directions frozen corn in well 4, Kroger canned corn in well 5, and Cadia canned corn in well 6. Here's my Cry1AB only gel. As you can see, there are only lines present for the DNA ladder and for bird's eye frozen corn. The extra line present in the bird's eye column is most likely due to a slightly high annealing temperature during PCR. Now here's the multiplex method gel. As you can see, all of the samples produced a 333 base pair segment, which results from the 16S RNA primers. Therefore, all of the samples had DNA in them. The bird's eye segment is a little bit bigger than the rest because it overlaps with the Cry1AB DNA segment. If you look closely, you can see a faint segment at 195 base pairs on all of the samples except the fresh corn. This segment is from the 35S CB primers. That all means that the only sample to contain the Bt gene was bird's eye frozen corn. However, all the samples except fresh corn contain the glyphosate gene, which protects corn against cauliflower mosaic virus. This discovery brings up an interesting point. Both Natural Directions frozen corn and Cadia canned corn are USDA certified organic, yet they possess a genetic modification. This genetic modification may not be the Bt toxin, but it's a modification all the same. In this case, organic doesn't mean non-GMO. More testing would be required to really see if the organic label holds any meaning when it comes to GMOs, but in this case, organic corn can still be genetically modified. So next time you're at the grocery store, think about what you're really buying.